This mRNA molecule has just come from transcription in the nucleus and is now in the cytoplasm. The 5' or GTP cap and poly A tail are not shown in this diagram. The mRNA is made up of multiple sequences of 3 nucleotides. Each sequence of 3 nucleotides is a triplet, or more specifically, a codon. For this video, the left side of the mRNA is the 5' end, and the right side is the 3' end. Now initiation begins. The large ribosomal subunit and the small ribosomal subunit come and attach to the mRNA. The blue molecule is an enzyme named aminoacyl tRNA synthetase. The 3 letters attached to the bottom of the tRNA make up the anticodon, which will match with the matching codon on the mRNA. The yellow pipe cleaner represents a tRNA molecule, which is a transfer RNA. The short end is the 5' end, and the taller end is the 3' end. The orange circle represents an amino acid. The amino acids are abbreviated to 3 letters. This amino acid is methionine. For initiation, a tRNA molecule and its matching amino acid get bound together by the enzyme aminoacyl tRNA synthetase. In the large ribosomal subunit, there is an A site, P site, and E site. The anticodon from the tRNA and the codon on the mRNA match due to the complementary base pairing rule. Now starts elongation. Another tRNA gets bound to its matching amino acid, leucine. The tRNA attaches at the A site in the ribosome. Now the ribosome shifts, and a peptide bond, the black pipe cleaner, forms between 2 amino acids. Now that methionine has bonded, the tRNA leaves the ribosome. The tRNA with leucine attached shifts to the P site. The process repeats again, this time with the tRNA matched with alanine. The tRNA attaches to the A site and the ribosome shifts while a peptide bond forms between leucine and alanine. The tRNA previously holding leucine now leaves the ribosome. The process repeats again for a tRNA matched with proline. Now, termination begins. Termination begins when the ribosome reaches a stop codon and the stop codon appears in the A site. Since there is no tRNA corresponding to the stop codon, a release factor binds to the A site. The binding of the release factor causes the polypeptide to be cleaved from the tRNA, and the ribosome, the tRNA, and the mRNA leave. Synthesis of the finished polypeptide is completed, and translation has ended. The new protein is ready for packaging and export. One disorder that results from a mutation is sickle cell anemia. For this demonstration, we will be focusing on the single codon in which the mutation occurs. In a normal cell, the normal DNA is transcribed into normal RNA. The codon codes for glutamic acid. The RNA is translated into the normal hemoglobin protein, which leads to the formation of a healthy red blood cell, which carries oxygen to the rest of the cell. But what causes sickle cell anemia? What causes this mutation is a substitution or point mutation in which one nucleotide base, adenine, gets replaced with a thymine base. This leads to a different RNA codon, which codes for a different amino acid, valine. Because of this change in amino acid, the resulting red blood cell is misshapen. As you can see, a sickle red blood cell is crescent-shaped, becoming rigid and sticky, which can slow or block blood flow. This leads to infection, pain, and fatigue.